Hello class, this is Mr. Miller. We're going to go over cellular processes. Homeostasis is the first process, and it is the process that our cells use to keep everything the same. So temperature regulation is an important aspect of homeostasis, and organisms like human beings sweat. Mammals sweat whenever they uh, get hot because the water that they sweat evaporates and takes energy away, and it reduces the amount of temperature that we have. Shivering is an uncontrolled contraction of muscles which burns fuel and creates heat. So shivering actually does warm us up. pH regulation is another aspect of homeostasis. pH regulation involves keeping the acid content of our blood constant at 7.4. Now, if we drink a Coca-Cola, it's going to have an acidic content of 2.5. It's very, very acidic. The lower number, the higher the acid. So this equation at the bottom of the page describes what happens when we drink hydrogen ions, which that's what acid is. It reacts with bicarbonate ions in our bloodstream to form bicarbonate, and then eventually that bicarbonate also forms water and carbon dioxide. So when you drink a Coke, the more carbon dioxide you're exhaling through your lungs, and that's the way that our body deals with the acid that a Coke has. Concentration of solutes is another homeostasis topic. We have a need for potassium in our cells, and we also have a need for sodium to be extruded or out of our cells. So we have evolved a potassium pump that allows us to kick out 3 sodium ions with the power or fuel of 1 ATP molecule. And it also takes in 2 potassium ions in the same breath, if you will, if you pardon my analogy anyway. And so when the sodiums occupy and are kicked out with the ATP fuel, it automatically opens up a spot for the potassiums to bind to, and then when they do, the conformational change creates a pathway for the potassiums to get back into the cell. There are also endoplasmic reticulum and Golgi apparatus organelles in our cells, and they help us to maintain status quo in each of our cells also. The endoplasmic reticulum produces protein. The Golgi apparatus uh, takes that protein that the ER produces and it postmarks it to wherever it has to go, so that if it's a cell that produces a lot of protein that is supposed to be excreted, the cell uses its Golgi apparatus to let every organelle know that this protein is going outside of the cell. And so it helps to maintain the amount of protein in each cell just by sending it to the places where it's supposed to go. The cell membrane is another aspect of cell process. The fluid mosaic model of the cell membrane indicates that there are proteins that span the entire breadth of the cell membrane, and they are involved in diffusion. They can allow molecules to get in, they allow molecules to get out. There are also other molecules that span the entire breadth of the membrane, but they're not involved in transport of molecules, they're involved in signal transduction. There are also peripheral proteins that are bound to either side that do have different functions. So this fluid mosaic model is a very important aspect of life. Note the cholesterol molecules that give this fluidity to the cell membrane. They allow suppleness and movement and folding, and it gives a little bit more movement to the cell membrane. This is a phospholipid, and this is the main constituent of the cell membrane. If you look at all these little molecules beside the proteins that make up this cell membrane, you can see that there are fatty areas that are hydrophilic. They don't like water. And there are also, on the other end of the molecule, hydrophobic areas where they do like water. The hydrophobic areas are the ones that are on the outside, the ones in contact with water. The hydrophobics, the fatty sides, are all associated together on the inside of this cell membrane. This potassium pump was talked about earlier, and we're going to do it again. The next topic is active and passive transport. And the potassium pump is an active transporter. It kicks out 3 sodiums and it brings in 2 potassiums And it does it all with the cost of one ATP molecule. And ATP is the universal fuel for every cell in our bodies. So without ATP, we wouldn't be able to exist. We wouldn't be able to function. And this is one of the main things that uses ATP, the sodium potassium pump, because all of our cells are kicking sodium out and taking potassium in to keep a homeostasis. Passive transport involves diffusion. Simple diffusion is the easiest or most simple way of understanding what passive transport is. If you get a huge concentration of molecules on one side of a membrane, after a while, if they're small enough, after time you will get an equal concentration on the inside and out. That is what simple diffusion is. Facilitated diffusion occurs when there's a carrier protein that allows molecules in or out. Now, we already talked about active transport, where energy is required from ATP to transport. Facilitated diffusion doesn't require any energy. It may require a ligand, which is like a key, and the key needs to open this carrier protein up so that diffusion can occur, but it doesn't cost any energy. And then we just looked at diffusion, or just simple diffusion. Here, osmosis needs to be discussed a little bit. This form of homeostasis involves, the cellular process involves a situation that's very analogous to what I've posted here as my illustration. You've got a beaker with a semi-permeable membrane in it, water molecules on one side, water molecules on the other side, but there's salt also. If you just add table salt to the permeable membrane, then this is what happens. The water molecules over here are bouncing around, and they bounce into the salt, so they can't get through the semi-permeable membrane as easily as the ones on the left can get to the right. So right to left isn't as easy as left to right, and this is what happens. The pressure, there's a higher pressure gradient, and it pushes up so that more of the solution is on the right hand side than the original solution on the left hand side. And that's a great way to describe exactly what goes on in our kidneys. The filtration that our kidneys undergo allows the big salt ions to be exposed through a semi-permeable cellular membrane, and so that allows this pressure to build up, and well, our blood pressure pushes back down so that the water can re-enter the left side, and the ions can get exported through our kidneys. Cell cycle interphase and mitosis are the cell cycle. Interphase is the stationary period where growth, synthesis, and preparation for division occur. Mitosis is both nuclear division and cytokinesis, so the nucleus divides and then the whole cell does. Meiosis occurs when there's nuclear division and cytokinesis, and then immediately after that there's another nuclear division, and you skip through the interphase that allows the protein or the DNA synthesis. So in the end result, meiosis involves daughter cells that only have half the amount of DNA that normal cells have. And these are normal cells that are undergoing mitosis, pointed to by the red arrows, and the interphase cells are pointed out by the blue arrows. Once again, interphase is a phase marked with DNA growth, replication, and other synthesis. Prophase is characterized when you can actually see the chromosomes forming. So when this cell begins to undergo mitosis, the first thing you see is the formation of chromosomes. And there are two chromosomes of each type. This picture doesn't show that well, but in metaphase are chromosomes, both chromosomes, and there should be two red, two blue, yellow, and green. They all line up on the equator, and then anaphase is when they start pulling apart. Telophase is after they've the nuclei, the chromosomes have all separated, and the nuclear membrane reforms around each of the cells, daughter cells, DNA. And then the cytokinesis is actually when the actual breakup of each of the cells occurs. And that is simple mitosis. Meiosis is in prophase, the stage that changes occur. So, as I said, meiosis is just like mitosis in that this phase, prophase 1, metaphase 1, anaphase 1, and telophase 1, are the same in both mitosis and meiosis. But in meiosis, instead of telophase going into interphase like mitosis and the DNA is replicated, in this case, the DNA in the cell is not replicated, and in a meiosis cell, and so the DNA that is created in interphase of mitosis is not created in meiosis. So you got a telophase identical to mitosis where you've got two daughter cells with a full complement, but then the two daughter cells in meiosis undergo an immediate division without reproduction of the DNA. And so this prophase is like the prophase 2 is where the chromosomes will appear thicker and they will form, and you'll be able to see them just like you would in mitosis. But in meiosis, there's only one of the chromosomes and one of the chromosome pairs. So when metaphase occurs, half of the chromosome is separated. These are chromatids. That's what's called chromatids. And when half of the DNA complement is pulled off, and in anaphase the chromosomes are pulled apart, and telophase is when the nuclear membrane forms around them, they're only half the DNA. And so the daughter cells, the gametes, only have half the DNA, and that's good because when they meet up with a gamete from a different organism in our species, the two unite and each half comes together to form a full complement of DNA for the daughters, for the zygote essentially. Growth, cell, and development. That's the next cell process. All organisms come from one cell. Everything that is an organism is due to differentiating cells from that one cell. And the difference, such as skin, muscle, brain cells, are all a result of differentiation. This differentiation results in, say, nerve cells. That's a good example. Multicellular organisms, different cell types perform different necessary and specific functions for the survival of the organism. So cell specialization results in nerve cells with dendrites that are receiving signals. And while every cell has a nucleus, every eukaryotic cell, but the signals picked up from dendrites are conducted down the axon. And the axon can be long. It can be up to a meter long for a cell. And in which case they need an insulator to keep the signal pure. And that's what myelin sheaths do. They really keep the signal pure so that by the time it gets down to the foot of this axon, it can reconnect with other dendrites or maybe a muscle cell for a contraction, or it just depends on what cell you're talking about. Red blood cells are responsible for carrying oxygen, and they've got a higher surface area because they're cupped. They have no organelles, so they can carry oxygen the best way that they can. Muscle cells are contractile, so they use proteins to do this, and they use a lot of energy, so there are a lot of mitochondria in each muscle cell. They produce and regulate a large amount of protein, so it's an important cell to have for our mobility. Epithelial cells form our skin. They also form the inside of our intestines. Epithelial cells are very, very useful, the most common cell in our body. They are involved in secretion, absorption, protection, transcellular transport, sensation detection, and they are permeability, selective permeability. They're very, very important. Other cells that are involved would be bone cells, white blood cells, fat cells, reproductive cells. All those cells are a result of cell specialization. So that's a cell process that's very important also.